Follow an alveolar duct along here and appreciate that the alveoli themselves are not parts of the walls of the ducts, but outpocketings from the walls, as you saw on the model. The wall itself is composed of little more than minimal bands of smooth muscle overlain by an almost invisible squamous epithelium. Here, the wall shows only a single smooth muscle cell cut transversely as a small bulge at the end of an alveolar wall. Alveolar sacs have the same basic structure and function as alveolar ducts. They are merely the somewhat expanded tip ends of the ducts, or outpocketings, as I've referred to before. Let me repeat what I said earlier, that the interalveolar septum is formed of two extremely attenuated sheets of the epithelial lining many capillaries and the epithelial lining of the air sacs. The many elongated nuclei belong largely to type 1 epithelial cells, or type 1 pneumocytes, and endothelial cells. It is not possible for you to distinguish between these two cell types. You can see many red blood cells in the capillary lumens, or perhaps in larger lumens of venules. The two other cell types located in the interalveolar septum or alveolar surface are type 2 epithelial cells, or type 2 pneumocytes, or septal cells, and macrophages. Type 2 cells are cuboidal and usually occur in the corners where two interalveolar walls come together. They produce surfactant. Picking out type 2 cells in hematoxin and eosin sections is not easy, but perhaps this is one over here, and perhaps another one here. Alveolar macrophages can be found anywhere. Once laden with dust particles, they then retire to a comfortable patch of connective tissue. Remember, you saw macrophages in sections of intrapulmonary bronchi I showed you earlier. This may be one here, as the little black dots indicate to me that they just may be dust particles that had been ingested by the macrophage.